Assalamualaikum. Today I will presenting of introduction the edible nest swiftlets, scientifically known as Aerodramus fucipagus. The topic is a unique avian species belonging to the Apodidae family. It is globally renowned for its extraordinary biological capability to construct nests entirely from its own hundred silica secretion, often referred to as the white gold of the top tropic. These nests are among the most expensive animal products in the world. About habitat and ecology, cave ecosystem, anthroponic structure, foraging ground. Cave ecosystem naturally inhabit dark limestone cave utilizing high frequency echolocation to navigate and nest in total darkness. And then structure important. Economic pillar, nutraceutical value and biological control. Economic pillar are multi-billion dollar export industry for Southeast Asia, providing high value income for rural and urban entrepreneurs. Alright.
So hi, so now I'm gonna explain about the number of the chromosome. The swiftlet or Aerodramus fucipagus has a 62 chromosome consisting of 40 macrochromosome and 22 microchromosome. This structure is typical for birds in the Apodidae family, where large and small chromosome plays a role in storing genetic information that control the physical characteristic and behavior of the swiftlet. So the total of the chromosome is 62 chromosome. 40 of it are macrochromosome and 22 of it are microchromosome. The sex chromosome are basically the same, XX for female and XY for male. So next is the core function of the swiftlet chromosome. The core function has 5: the genetic storage, inheritance, cell division, sex determination, and evolutionary adaptation. For the genetic storage, they hold the DNA, encodes traits such as feather structure, flight ability, and nest building behavior. For the inheritance, the chromosome ensure that offspring inherits characteristic from parent swiftlet, maintaining species identity. For the cell division, during mitosis and meiosis, chromosome guarantee accurate distribution of genetic material, supporting growth and reproduction. For the sex determination, in swiftlet, sex is determined by Z and W chromosome. For the female, it is Z and W. For the male, it's gonna be Z and Z. Gliding reproductive rules. For the evolutionary adaptation, the balance of an microchromosome and 22 microchromosome supports genetic diversity, helping swiftlet adapt flight, demand, and ecological niches. So that's.
Aerodramus fucipagus, or the edible nest swiftlet, has a genome size of approximately 1.13 gigabase, which is equivalent to about 1126.08 megabase. Its genome size is considered relatively compact in this typical of bird species. In addition, the genome contains around 21,285 predicted protein coding genes. These genes were identified through genome sequencing and annotation study. The term predicted genes mean that the genes were identified using computation analysis and may not all have been experimentally verified. Overall, The genome size and genes number of Aerodramus fuchsivagus are consistent with the characteristic commonly found in other bird species.
Hello everyone, my name is Susna. I will present about the gene and protein product of the swiftlet, also known as the bird's nest swiftlet. This topic that I focuses on how a specific gene produces a protein that helps the bird build its nest. Swiftlets are unique birds because they build their nest using their saliva. These nests are not made from twigs like other birds, but from a special protein found in their saliva. This process is controlled by specific genes inside their bird's body. The gene involved is called the mucin gene, specifically MUC5AC or MUC5B. These genes are responsible for producing a substance called mucin. They are found in the salivary glands of the swiftlet. The protein produced is called mucin, which is a type of glycoprotein. It has special properties which are sticky and viscous. When exposed to air, this protein can harden and form a solid structure. The mucin proteins plays an important role in nest building. First, it forms the structure of the bird's nest. Second, it acts as a natural adhesive to stick the nest into walls. It also helps protect the eggs and baby birds. In conclusion, the MUC5AC or MUC5B genes produce mucin protein. This protein is very important because it allows the swiftlet to build its nest. Without this gene and protein, the nest cannot be formed. That's all from me.
Swiftlets are a small bird that produce one of the most valuable natural products known as edible bird nest or EBN. They create their nest using only their saliva, which harden after exposure to the air. The formation of this nest is controlled by specific genes found in the salivary glands, such as CREB3L2, and pathways like EGFR and MPAK. These genes play an important role in producing Munchkin's glycoprotein, a substance that makes the nest sticky, strong, and able to hold in structure. Interestingly, the expression of these genes is not fixed. It can be influenced by environmental factors such as bird diet and the quality of the habitat. This means better living conditions can affect the quality of the nest produced. Edible bird nest, widely used in many industries. In the food industry, it is consumed as health tonic. In cosmetics, it is commonly added into skincare because it believed that help with anti-aging and skin repair. Research also that EBN may help support the immune system that promote wound healing, making it valuable and nutritional field. By studying swiftlet genes, scientists can better understand how saliva proteins are produced and how biomineralization happens. This knowledge is useful not only for biology but also for improving commercial bird nest farming. Overall, genetic research on swiftlets can help increase production efficiency and improve the quality of the edible bird nest in the future.
Based on the study's review, several important findings were identified. First, the genome of the edible nest swiftlet Aerodramus fuchsivagus was successfully sequenced for the first time. The study identified genes and protein involved in edible bird nest production, providing valuable information on its nutritional, medicinal, and biotechnological potential. Second, research on cystic fibrosis highlighted the potential of RNA and DNA-based therapies, including mRNA therapy, gene addition, and gene editing. These approaches aim to restore CFTR function and could potentially treat all types of CF mutation, although efficient delivery to target cell remain a challenge. Third, a study on edible bird nests showed that heat treatment significantly improve its solubility and water holding capacity. Higher temperature modified the protein structure, allowing the nest to absorb more water and increasing its functional for food and nutraceutical application. In conclusion, the edible nest swiftlet has a genome size of approximately 1.13 gigabase, containing about 21,285 protein-coding genes. Important machine genes such as MAC5AC and MAC5B are a possibility for producing the glycoproteins that from edible bird nests. Besides their biological significance, edible bird nests have considerable economic valuable and widely used in the food, cosmetic, and health supplement industry. Understand the swiftlet genome not only supports sustainable bird nest farming but also provide useful insight for future genetic and biomedical research.